Hello, and thank you for joining me on this gentle journey. Tonight, we drift along quiet Australian rivers, where the platypus moves beneath the surface in calm twilight water. In this soft river world, everything feels balanced between stillness and gentle motion as the current flows slowly through reeds and reflections of fading light. The platypus carries a quiet wonder, an animal that seems almost impossible, yet exists in perfect harmony with its environment, gliding through water with peaceful ease. Let your mind settle into this calm river scene, where everything feels soft, warm, and unhurried. If you enjoy this relaxing moment, please consider liking, subscribing, or sharing your thoughts below. But for now, simply breathe and relax. Let's begin. In the quiet river systems of eastern Australia, water moves with a patient, unhurried rhythm, shaping landscapes that appear still on the surface, but are constantly shifting beneath it. Eucalyptus forests lean over the banks, their roots gripping into damp soil, while their canopies scatter sunlight into fragmented patterns across the water. The air carries a faint dryness, even above the humidity of the river corridor, a reminder of how extreme seasonal cycles define this land. Rain arrives in sudden bursts, transforming narrow creeks into expanding channels that reshape their own boundaries. Between these cycles, heat tightens the ground and reduces the waterways to slower, more concentrated flows. Below the surface, the river system supports a dense and often unseen network of life, where aquatic insects cling to submerged vegetation and crustaceans move through the sediment in slow, deliberate patterns. Small fish navigate the currents in tight coordination, responding instantly to changes in pressure and vibration. Each organism occupies a narrow ecological role shaped by water level, temperature, and oxygen availability. During dry periods, many species retreat into deeper pools, while wet seasons redistribute nutrients and reshape the balance of survival. The entire system functions like a living circuit, where every fluctuation in flow alters the behavior of countless interconnected forms of life. It is within this shifting environment that the platypus moves quietly, rarely revealing itself above the surface for long. Its presence is most often inferred rather than directly observed, marked by subtle disturbances in the water or faint impressions along muddy banks. The animal navigates submerged tunnels and shallow channels with a precision shaped by long evolutionary adaptation. To an outside observer, it seems almost unreal, appearing only briefly before disappearing again into the complexity of the river. Yet within this habitat, it is a consistent and deeply integrated presence, moving through cycles of feeding and rest that align with the rhythm of the water itself. The rivers of eastern Australia are among the oldest freshwater systems on Earth, shaped by continental drift and long periods of geological stability. Unlike younger river networks that frequently shift and reorganize, these waterways have maintained recognizable courses for immense stretches of time. This ancient stability has allowed unusual evolutionary pathways to persist, preserving lineages that might otherwise have disappeared. The surrounding landscape reflects this deep history, with weathered rock formations and soils that have been reworked by countless cycles of erosion and deposition. Within this setting, life has adapted not through rapid expansion, but through careful specialization. In these waters, survival depends on sensitivity rather than strength. The platypus closes its eyes, ears, and nostrils as it dives, relying instead on electroreception to interpret the invisible signals of prey beneath the sediment. Its movements are slow and deliberate, designed to minimize disturbance in the water column. Each sweep of its bill across the riverbed becomes a form of mapping, translating electrical impulses into a navigational understanding of its surroundings. In this submerged darkness, perception is not visual, but entirely tactile and electrical, forming a unique sensory experience unlike that of most mammals. It is within this combination of ancient geography and finely tuned adaptation that the platypus finds its place. The river does not demand dominance, only persistence, and the platypus embodies this principle through its quiet existence. Every movement, every dive, every return to the burrow is part of a system shaped by time rather than intention. In these quiet waters, evolution is not a dramatic force, but a slow accumulation of small adjustments, each one layered upon the last. The story of the platypus begins here, not as an exception, but as a continuation of nature's long and intricate unfolding. Across the early history of European natural science, few encounters with Australian wildlife created as much confusion as the first preserved specimens of the platypus. When descriptions and skins arrived in Britain during the late 18th century, many naturalists assumed the material had been fabricated. The combination of features appeared incompatible with known biological categories, as if several unrelated animals had been assembled into a single form. A bill resembling that of a waterfowl, a tail reminiscent of a beaver, and limbs suited for swimming, created immediate skepticism amongst scientists who relied on established classification systems. Scientific scrutiny intensified as more specimens became available, confirming that the animal was genuine rather than artificial. Careful anatomical study revealed that every unusual trait served a functional role rather than existing as decorative oddity. The bill, although similar in appearance to that of a duck, was structurally different and contained specialized sensory tissue. The tail stored fat reserves, providing energy during periods of reduced food availability. The limbs were adapted for aquatic propulsion, with webbing that could retract when moving across land. Each feature reflected adaptation to a specific ecological niche rather than randomness. Biologists eventually placed the platypus within a group known as monotremes, one of the most ancient branches of mammalian evolution. Members of this group share a reproductive strategy that differs significantly from most mammals. They lay eggs rather than giving live birth. This characteristic links them to earlier stages of vertebrate evolution, while still retaining key mammalian traits such as milk production. The coexistence of these features within a single organism challenged existing definitions of what a mammal could be. Further investigation revealed additional biological traits that deepened the scientific intrigue. Male platypuses possess venom-producing glands connected to spurs located on their hind limbs. This venom is not used for predation, but plays a role in competition and defense, particularly during reproductive periods. The presence of venom in a mammal is extremely rare, adding another layer of complexity to the animal's classification. Although not typically dangerous to humans, the venom can cause severe pain and prolonged discomfort, indicating its effectiveness within species-specific interactions. Reproductive biology in the platypus also differs from that of most mammals. Females lay eggs that are incubated in burrows constructed along riverbanks. After hatching, the young are not fed through nipples, as no such structures exist. Instead, milk is secreted through specialized skin regions and absorbed by the offspring. This method of nourishment represents a unique variation of mammalian lactation, demonstrating that milk production can exist independently of mammary nipples. The young remain dependent on the mother for an extended period while developing the ability to forage independently. Modern genetic analysis has confirmed that monotremes diverged from other mammalian lineages hundreds of millions of years ago. This extended separation explains the presence of traits that appear primitive when compared with placental and marsupial mammals. However, these characteristics are not outdated remnants, but successful adaptations that have persisted in stable ecological conditions. The Australian continent, isolated for long periods of geological time, provided an environment where such evolutionary pathways could continue without interruption from competing lineages. Within this framework, the platypus emerges not as an anomaly, but as a representative of an ancient evolutionary branch that followed a different trajectory. Its combination of traits reflects a long history of adaptation to freshwater environments, where survival depends on sensory precision, energy efficiency, and reproductive resilience. The initial scientific confusion surrounding the species gradually gave way to recognition of its significance, not as an error in classification, but as evidence of the diversity possible within mammalian evolution. At the surface level, the platypus bill resembles a soft, duck-like structure, but beneath that outward appearance lies one of the most specialized sensory organs among mammals. Early anatomical studies assumed it functioned primarily as a tool for feeding or digging. Yet closer examination revealed a dense network of nerve endings embedded within its flexible tissue. This discovery shifted scientific understanding of how the animal interacts with its environment, showing that the bill is not simply a physical extension, but a highly refined sensory interface. Within the structure of the bill, thousands of mechanoreceptors and electroreceptors work together to interpret subtle environmental signals. These receptors detect both physical disturbances in the water and minute electrical fields generated by muscular activity in aquatic organisms. Worms, insect larvae, and small crustaceans all produce faint bioelectric signatures during movement, and the platypus is able to register these signals even when visual information is completely absent. The distribution of sensory cells across the bill surface allows for directional sensitivity, enabling the animal to determine the location of prey with remarkable precision. During foraging, the platypus undergoes a striking sensory shift. As it submerges, the eyes close, the ears seal, and the nostrils compress, effectively removing reliance on conventional senses that depend on light or airborne sound. In this state, the animal navigates entirely through tactile and electrical input gathered by the bill. Movement becomes deliberate and measured, with slow sweeps across the riverbed that minimize disruption to sediment layers. Each motion serves to expand the sensory field, creating a continuous stream of information from the surrounding environment. Beneath layers of mud and gravel, hidden organisms generate weak electrical fields as they contract and move. The platypus detects these signals and translates them into spatial awareness, allowing it to distinguish between inert objects and potential prey. Even in turbid water, where visibility is non-existent, it can locate targets with high efficiency. This ability is not dependent on sight or sound, but on an entirely different sensory modality that interprets the electrical properties of living tissue. The riverbed, in this context, becomes a dynamic map of biological activity. Scientific research has shown that the platypus brain contains specialized processing regions dedicated to interpreting signals from the bill. Neural pathways prioritize electroreceptive input during underwater foraging, effectively reallocating cognitive resources to enhance detection accuracy. This adaptation demonstrates a level of sensory integration that is rare among mammals, but more commonly observed in certain fish and amphibian species. The convergence of such traits across distant evolutionary lineages highlights the repeated emergence of electroreception as an effective survival strategy in aquatic environments. From an evolutionary perspective, the development of electroreception in a mammal represents a significant divergence from typical sensory systems. Most terrestrial mammals rely heavily on vision, hearing, and olfaction, yet the platypus operates within a framework that prioritizes electrical and mechanical cues. This shift reflects adaptation to freshwater habitats where light penetration is limited and prey often remains concealed beneath sediment. Over long periods of isolation in Australia's river systems, this sensory specialization became increasingly refined, allowing the species to exploit ecological niches with minimal competition. Ongoing scientific investigation continues to refine understanding of how the bill functions at both structural and neurological levels. Research suggests that the integration of electroreception with tactile sensing creates a unified perceptual system that is highly efficient in low-visibility aquatic environments. Rather than relying on multiple independent senses, the platypus merges input into a cohesive representation of its surroundings. This adaptation illustrates how evolution can produce entirely novel sensory solutions when environmental conditions favor alternative modes of perception. Among the most unexpected biological features found in any mammal, the male platypus carries a pair of sharp spurs located on the inner ankles of its hind limbs. These structures are connected to crural glands that become active during the breeding season, producing a complex venom that is delivered through a groove in each spur. Unlike venom systems used for hunting in reptiles or arachnids, this mechanism serves a different role entirely, shaped by intraspecies competition rather than predation. Examining the chemical composition of platypus venom reveals a mixture of peptides and proteins that affect pain pathways in mammals. The resulting sensation is known to be extremely intense, often described in clinical observations as severe and long-lasting. While not typically life-threatening to humans, the pain does not respond well to standard analgesics, which makes it particularly notable in medical case reports involving accidental encounters. The persistence of discomfort suggests that the venom targets neurological pathways in a highly specific manner. During mating season, hormonal changes trigger increased activity in the venom glands of males. This period is marked by heightened territorial behavior as individuals compete for access to receptive females. Encounters between males can involve aggressive physical interactions, during which the spurs may be used as defensive or competitive weapons. The venom likely evolved as a mechanism to establish dominance and reduce physical conflict duration by discouraging repeated engagement. From a structural standpoint, the spur itself is composed of keratin, similar to fingernails and claws in other mammals, but it is uniquely adapted for venom delivery. The associated gland connects through a duct system that allows venom to flow into the spur when activated. This arrangement is rare among mammals and represents one of the few known examples of a functional venom apparatus in the group. Its presence highlights the diversity of evolutionary strategies, even within closely related species. Field observations of wild platypuses indicate that venom use is strongly seasonal and closely tied to reproductive cycles. Outside of breeding periods, the glands reduce activity and aggressive interactions become less frequent. This seasonal regulation suggests that the venom system is not intended for everyday survival, but is instead reserved for specific biological contexts. In this way, it functions as a specialized tool shaped by reproductive pressures rather than general defense needs. Comparative studies with other venomous animals show that the platypus system is independently evolved, not derived from a shared ancestral venom mechanism. This independent emergence of venom production demonstrates how similar environmental pressures can lead to the development of comparable biological solutions across unrelated lineages. In the case of the platypus, isolation within Australian freshwater ecosystems provided conditions that allowed this trait to persist and refine over time. Despite its unusual nature, the venom system does not define the platypus as a dangerous animal in the broader ecological sense. Encounters with humans are rare, and the species remains largely reclusive in its habitat. Its venom is more significant as a subject of scientific study than as a widespread ecological threat. Researchers continue to investigate its biochemical properties, both to understand its evolutionary origin and to explore potential insights into pain mechanisms in mammals. Within the broader biological profile of the platypus, the presence of venom adds another layer of complexity to an already unusual organism. It exists alongside egg-laying reproduction, electroreceptive sensing, and milk production without nipples, forming a combination of traits rarely seen in a single mammalian lineage. Each feature reflects a distinct evolutionary response to environmental and reproductive pressures within Australia's freshwater systems, reinforcing the species' role as a living example of diverse adaptive strategies. Along the edges of eastern Australian waterways, the riverbanks hold an intricate network of tunnels that reveal far more about platypus life than surface observation ever could. These burrows are excavated into soft soil above the waterline, carefully positioned to remain stable during fluctuations in river height. Their entrances are often concealed beneath roots, vegetation, or natural overhangs, reducing exposure to predators and environmental disturbance. From the outside, they appear unremarkable, yet internally they form complex shelter systems shaped by repeated use across seasons. Deep within these earthen structures, the platypus spends a significant portion of its life resting, grooming, and avoiding external conditions that fluctuate above ground. The temperature and humidity inside the burrow remain more stable than the surrounding environment, offering a controlled space for energy conservation. Multiple chambers may exist within a single tunnel system, including nesting areas and resting zones, separated by narrow passages. This architecture reflects both instinctive behavior and long-term adaptation to riverine instability. During breeding periods, female platypuses modify their burrows by sealing entrances with soil and plant material, creating a more secure internal environment. Within these enclosed chambers, egg-laying occurs, marking one of the most distinctive reproductive traits among mammals. The eggs are soft-shelled and incubated in a carefully regulated microenvironment maintained by the mother's body positioning and nesting materials. This stage of development remains hidden from external observation, occurring entirely within the protected confines of the burrow system. After hatching, the young emerge in a highly dependent state, lacking fur and requiring constant care. Instead of nipples, the mother secretes milk through specialized skin regions, which pools on the surface and is absorbed by the offspring. This feeding method is unique among mammals and requires the young to remain close to the mother for sustained nourishment. Growth during this period is gradual, supported by frequent feeding and protection within the burrow chamber. Outside the nesting phase, burrows continue to function as resting sites and refuges from environmental stresses. Flooding events can temporarily inundate entrances, but deeper sections often remain intact, allowing the platypus to survive periods of high water. During dry cycles, these same structures provide protection from heat and reduced surface water availability. The durability of burrow systems is therefore essential to long-term survival in a habitat defined by fluctuation. Field studies have shown that platypus burrows can extend several meters in length, with branching tunnels that serve different purposes. Some lead directly to water access points, while others function as escape routes or secondary resting chambers. The arrangement is not random, but reflects repeated refinement through occupation over time. Each burrow represents a record of behavioral adaptation to local environmental conditions. Ecologically, the relationship between platypuses and riverbanks extends beyond shelter alone. The act of digging contributes to soil turnover and can influence local microhabitats used by invertebrates and small aquatic organisms. In this way, burrow construction plays a subtle role in shaping the immediate environment, linking the animal's behavior to broader ecological interactions within the river system. Across generations, these hidden structures anchor the species to specific stretches of water, reinforcing a life cycle closely tied to stability in freshwater habitats. Even as surface conditions change with rainfall and seasonal variation, the underground architecture remains a consistent feature, supporting reproduction, protection, and survival. Within these concealed spaces, much of the platypus life cycle unfolds away from direct observation, yet remains essential to the continuity of the species in its natural range. Genetic and fossil evidence places the platypus within one of the oldest surviving branches of mammalian evolution, belonging to the Monotreme lineage that diverged from other mammals more than 160 million years ago. This separation occurred during a period when early mammals were still diversifying from reptile-like ancestors, long before placental and marsupial groups became dominant. As a result, monotremes retain a mixture of ancestral traits alongside highly specialized adaptations that reflect millions of years of independent evolutionary history in the southern hemisphere. Paleontological discoveries across Australia and South America have helped reconstruct the deep origins of this lineage, revealing that early monotreme relatives once had a broader distribution across ancient supercontinents. Over time, continental drift and changing climates restricted their range, leaving Australia as the primary refuge for surviving species. This geographic isolation created conditions where evolutionary experimentation could continue without direct competition from many other mammalian groups, allowing unusual traits to persist and diversify. Anatomical comparisons between monotremes and other mammals highlight a mosaic of characteristics that reflect both ancient and derived features. The presence of egg-laying reproduction, for example, contrasts sharply with the live birth seen in marsupials and placental mammals. At the same time, monotremes share core mammalian traits, such as hair, warm-blooded metabolism, and the production of milk for offspring nourishment. This combination places them at a unique intersection in vertebrate evolution, bridging characteristics associated with both earlier and more derived lineages. Molecular studies have provided further clarity on the evolutionary timeline, indicating that monotremes split from therian mammals well over 160 million years ago. Since that divergence, their lineage has followed a separate evolutionary path shaped by environmental pressures specific to Australia and New Guinea. This long period of isolation has resulted in a suite of biological features not seen in other modern mammals, including electroreception in the platypus and specialized reproductive strategies that differ fundamentally from those of most living species. Comparative embryology has also contributed to understanding how monotreme development differs from other mammalian groups. During early development, platypus embryos exhibit a combination of traits that reflect both reptilian and mammalian characteristics, particularly in skeletal and reproductive structures. These developmental patterns provide insight into how complex biological systems evolve incrementally over time, with existing structures modified rather than replaced entirely. Such findings help clarify the gradual transitions that occurred during early mammalian evolution. Evolutionary biology views the platypus as a living representation of a lineage that preserves information about early mammalian diversification. Its genome contains a mixture of genes associated with reptilian ancestry and those linked to mammalian innovations such as lactation and endothermy. This genetic composition offers researchers a valuable window into how new biological functions emerge and stabilize within populations over extended periods. Rather than being a transitional form, it represents a fully adapted species that reflects a distinct evolutionary pathway. Within modern ecosystems, the platypus continues to occupy freshwater habitats that resemble, in some respects, the environments that shaped its ancestors. Rivers, streams, and floodplain systems provide stable ecological niches where its specialized traits remain advantageous. Its survival demonstrates how ancient lineages can persist alongside more recently evolved mammals, maintaining continuity across vast spans of geological time. In this way, the platypus serves as a biological record of deep evolutionary history preserved within a single living organism. As daylight fades across eastern Australian waterways, the activity of many aquatic species shifts in response to changing light levels and temperature gradients within the river system. The platypus becomes increasingly active during these hours, emerging from its burrow to begin extended foraging sessions that can last several hours each night. This timing reduces direct competition with many diurnal predators and aligns feeding activity with the behavior patterns of prey species that move through the water column after dusk. Emerging from the riverbank entrance, the animal enters the water with minimal surface disturbance, immediately transitioning into a highly specialized hunting mode. The eyes close almost completely, the ear openings seal, and the nostrils compress, removing reliance on vision, hearing, and smell while submerged. This anatomical adjustment redirects sensory focus toward mechanoreception and electroreception, allowing the platypus to navigate and detect prey using signals generated within the aquatic environment itself. Beneath the surface, movement becomes highly controlled and energy efficient. The forelimbs paddle in a steady rhythm, while the hind limbs and tail provide stabilization and directional control. Rather than pursuing prey through rapid chases, the platypus follows subtle electrical cues produced by small organisms embedded in sediment or drifting near the riverbed. These cues are interpreted through specialized receptors located in the bill, which continuously scan the environment during foraging. Throughout each hunting session, the bill sweeps laterally across the riverbed in repeated arcs, sampling a wide area of sediment and submerged debris. This motion allows the animal to construct a sensory map of its surroundings based on variations in electrical and mechanical input. Worms, insect larvae, and small crustaceans become detectable even when buried beneath layers of silt, as their muscle contractions generate faint bioelectric fields. The ability to detect such signals provides a significant advantage in environments where visibility is limited. Energy expenditure during feeding is carefully balanced against intake requirements, as the platypus must consume a substantial amount of food relative to its body mass to sustain its metabolic needs. Each foraging session is therefore structured around efficiency, with minimal wasted movement and a strong reliance on sensory accuracy. Periods of intense activity are followed by brief pauses within the water column or along the riverbed, allowing for recalibration of sensory input before continuing. Field observations have shown that feeding behavior is influenced by seasonal variations in water flow and prey availability. During periods of higher rainfall, increased water movement redistributes aquatic organisms, altering feeding patterns and requiring adjustments in foraging strategy. In contrast, lower water levels concentrate prey into smaller areas, which can increase feeding efficiency but also intensify competition with other aquatic species. The platypus adapts to these fluctuations through flexible movement patterns and sustained reliance on electroreceptive hunting. As the night progresses toward early morning, foraging activity gradually decreases, and individuals return to their burrows along the riverbank. The transition back to land involves careful navigation to locate concealed entrances, often guided by memory of spatial landmarks and water flow patterns. Once inside the burrow, the animal enters a state of reduced activity, conserving energy until the next feeding cycle begins. This repeated rhythm of movement between water and shelter defines much of its daily existence and ensures survival in an environment shaped by constant variation. Across the freshwater systems of eastern Australia and Tasmania, the survival of the platypus is closely tied to the stability and quality of river environments that are increasingly shaped by human activity and climatic variation. These waterways form interconnected habitats where water flow, sediment structure, and prey availability determine whether populations can persist in a given region. Unlike many adaptable mammals, the platypus depends on a narrow ecological balance between aquatic and terrestrial conditions, making it especially responsive to even subtle environmental changes. Hydrological modification through dam construction, water extraction, and river regulation has altered the natural variability of many catchments where platypuses once moved freely. Changes in flow regimes can reduce seasonal flooding events that normally maintain healthy riverbank structure and redistribute nutrients throughout the ecosystem. When water levels become artificially stabilized or heavily diverted, the natural cycles that shape burrow stability and prey distribution are disrupted, leading to long-term impacts on habitat suitability. Fragmentation of river systems also limits movement between populations, reducing genetic exchange across regions. Water quality represents another critical factor influencing platypus survival. Agricultural runoff, urban development, and industrial activity can introduce pollutants into freshwater systems, affecting both the animals directly and the invertebrate communities they rely on for food. Because the platypus feeds on a wide range of aquatic larvae, worms, and crustaceans, reductions in invertebrate abundance can quickly translate into nutritional stress. Sediment changes caused by erosion or land clearing may also alter riverbed composition, reducing the availability of suitable foraging zones where prey is normally concentrated. Extended periods of reduced rainfall and increased evaporation linked to climate variability place additional pressure on freshwater ecosystems. Lower river volumes can lead to isolated pools, fragmented habitats, and reduced oxygen levels in stagnant water. During these conditions, prey populations may decline or become unevenly distributed, forcing platypuses to travel further distances to meet energy requirements. Conversely, extreme rainfall events can produce sudden flooding that damages burrow systems, destabilizes riverbanks, and temporarily displaces individuals from established territories. Ecological studies have shown that the platypus functions as an indicator species for freshwater health, meaning that its presence or absence often reflects the broader condition of the ecosystem. Because it depends on both aquatic invertebrate abundance and intact riverbank structures, changes in its population density can signal shifts in environmental quality across entire catchments. Monitoring programs use sightings, burrow activity, and genetic sampling to track population trends and identify regions under environmental stress. This makes the species an important reference point in freshwater conservation research. Riparian zones, where land meets water, play a central role in maintaining suitable habitat for the platypus. Vegetation along riverbanks stabilizes soil, reduces erosion, and provides cover for burrow entrances. When these areas are cleared or degraded, banks become more susceptible to collapse during high-flow events, potentially destroying nesting sites. The integrity of these transitional zones therefore influences both reproductive success and long-term habitat persistence. In many regions, restoration efforts focus on replanting native vegetation and reducing livestock access to river edges to improve bank stability. Conservation assessments have classified the platypus as a species of concern in several parts of its range, particularly in areas experiencing rapid environmental change. While it is not globally extinct or critically endangered, localized declines have been recorded in regions where river systems have been heavily modified or degraded. Conservation strategies emphasize maintaining connected waterways, protecting water quality, and preserving natural flow variability. These measures aim to support not only the platypus, but also the broader freshwater ecosystems on which many other species depend. Long-term survival of the species is closely linked to the maintenance of natural hydrological cycles and intact river networks. As environmental pressures continue to evolve, ongoing research and monitoring remain essential for understanding how populations respond to changing conditions. The platypus, deeply embedded within freshwater systems, reflects the health of entire aquatic landscapes, making its persistence dependent on the careful balance between human activity and ecological integrity across Australia's river corridors. Scientific interest in the platypus has persisted for centuries because it occupies a position in biology that resists simple categorization. When early naturalists first encountered the species, they attempted to place it within existing frameworks that divided animals into mammals, birds, and reptiles. The combination of traits observed in the platypus did not fit comfortably within any single group, leading to extended debate and repeated reassessment as new anatomical and physiological data became available. Modern biological classification places the platypus within Monotremata, a distinct mammalian order that diverged early from other mammalian lineages. This placement reflects a synthesis of genetic, fossil, and anatomical evidence accumulated over time. Despite its classification as a mammal, the species retains characteristics that are uncommon among most living mammals, including egg-laying reproduction and specialized sensory adaptations not found in placental or marsupial groups. These features continue to challenge assumptions about how mammalian traits are defined and distributed. Genomic sequencing has provided deeper insight into the evolutionary history of the platypus, revealing a mosaic of genetic elements associated with both ancestral vertebrate systems and more derived mammalian functions. The genome contains genes linked to reptilian-like egg development, alongside those responsible for mammalian lactation and thermoregulation. This combination illustrates how evolutionary pathways can preserve ancient biological systems while simultaneously developing new functional traits. Rather than representing a transitional form, the platypus demonstrates a stable and highly specialized evolutionary outcome. Comparative physiology has further highlighted the uniqueness of this species. The presence of electroreception, venom production in males, and milk secretion without nipples represents a convergence of traits that are rarely observed together in a single organism. Each of these systems operates independently, yet contributes to the overall survival strategy of the animal. Electroreception enhances feeding efficiency in turbid water. Venom plays a role in reproductive competition, and lactation ensures offspring development in protected burrow environments. Fossil evidence of early monotreme relatives has helped contextualize the evolutionary trajectory of the platypus. Ancient species such as Steropodon and Obdurodon, discovered in Australian sedimentary deposits, show that monotreme-like animals have existed for tens of millions of years. Some of these extinct relatives possessed more generalized dental structures, suggesting different feeding strategies compared to modern platypuses, which lack functional teeth in adulthood. These findings indicate a long history of adaptation to freshwater environments with shifting ecological pressures. Evolutionary biology interprets the platypus as an example of long-term lineage persistence in relative geographic isolation. The separation of Australia from other landmasses allowed unique evolutionary experiments to unfold without significant external competition from other mammalian groups. Over extended periods, this isolation contributed to the retention and refinement of traits that might otherwise have been lost or replaced in more competitive ecosystems. The result is a species that reflects both ancient heritage and ongoing adaptation. Behavioral studies have added another dimension to understanding how this animal functions within its environment. Despite its unusual anatomy, the platypus exhibits highly efficient foraging behavior, careful energy management, and strong site fidelity to established river systems. Its interactions with the environment are shaped by sensory input rather than visual dominance, demonstrating how alternative perceptual systems can support complex survival strategies. These behavioral patterns reinforce the idea that evolutionary success is not dependent on conformity to a single biological template. Philosophical discussions within evolutionary science often reference the platypus when addressing the limits of classification systems. Its existence demonstrates that biological diversity cannot always be neatly organized into rigid categories without losing important nuance. Instead, it highlights the importance of flexible frameworks that accommodate exceptions and overlapping traits. Each new discovery about the species expands scientific understanding of how life can evolve under different environmental and historical conditions. Across the evolutionary history of Earth, few animals have challenged human classification systems as persistently as the platypus, a species whose biology reflects a combination of traits that defies simple categorization. Its existence within Australian freshwater ecosystems provides a living example of how evolutionary pathways can produce organisms that do not conform to conventional expectations of form or function. Rather than fitting neatly into established categories, it occupies a position that expands them, demonstrating that biological diversity often extends beyond structured frameworks. Early scientific encounters with the species reveal a recurring pattern of uncertainty, followed by gradual acceptance. When the first preserved specimens reached Europe, they were met with skepticism due to their unusual combination of characteristics. Over time, repeated anatomical study confirmed that the animal was authentic, prompting revisions in biological classification systems. This shift illustrates how scientific understanding evolves in response to new evidence, particularly when that evidence does not align with existing assumptions about how mammals should appear or behave. In ecological terms, the platypus represents a specialized adaptation to freshwater environments that vary significantly across seasons and geographic regions. Rivers and streams in eastern Australia provide habitats where fluctuations in water flow, sediment composition, and prey availability shape survival strategies. Within these systems, the platypus has developed a suite of traits that support efficient foraging, reproduction, and energy management under conditions that are often unpredictable. These traits are not isolated features, but interconnected components of a broader biological system. Reproductive biology in the species highlights one of its most distinctive characteristics. Egg-laying in mammals is an extremely rare trait, confined to monotremes, and represents an evolutionary link to earlier vertebrate lineages. After hatching, offspring rely on milk secreted through specialized skin glands, a form of lactation that operates without nipples. This method of nourishment ensures that young remain protected within burrow environments during early development, where external conditions are more stable than surface habitats. The combination of egg incubation and lactation reflects a unique reproductive strategy not found in other mammalian groups. Sensory adaptation further distinguishes the platypus from most mammals. The electroreceptive capabilities of its bill allow it to detect electrical signals generated by the movement of aquatic organisms beneath sediment. This ability enables efficient foraging in environments where visibility is limited by turbidity or low light conditions. By integrating mechanical and electrical sensory input, the animal constructs an internal representation of its surroundings that is fundamentally different from vision-based perception systems common in other mammals. Physiological studies also highlight the presence of additional specialized traits, including venom production in males and highly efficient energy storage mechanisms in the tail. The venom system, active primarily during reproductive competition, reflects selective pressures associated with mating behavior rather than predation. Energy storage in the tail supports survival during periods when food availability fluctuates, particularly in environments influenced by seasonal variation in water flow and prey abundance. These adaptations demonstrate how multiple biological systems can evolve in response to different ecological demands. From an evolutionary perspective, the platypus serves as a representative of long-term lineage persistence in geographically isolated environments. The separation of Australia from other continents allowed monotreme lineages to develop independently over extended periods of geological time. This isolation reduced direct competition with other mammalian groups, enabling the retention of ancient traits alongside the development of new specialized functions. As a result, the species reflects both continuity with early mammalian evolution and adaptation to modern ecological conditions. Scientific research continues to explore how the combination of traits observed in the platypus emerged through incremental evolutionary changes. Rather than appearing suddenly, each characteristic is understood as part of a gradual modification of existing biological structures. Electroreception, lactation, egg-laying, and venom production each represents separate evolutionary developments that became integrated into a single organism over long timescales. This integration illustrates the flexibility of evolutionary systems in producing diverse solutions to environmental challenges. Further comparative studies with other vertebrate groups help clarify how similar adaptations can arise independently in different evolutionary lineages facing comparable ecological pressures. By examining genetic pathways and developmental biology, researchers can trace how small functional shifts accumulate into complex traits. The platypus therefore serves as a key reference in understanding how modular biological systems evolve, where individual components can be repurposed and refined without requiring complete redesign of an organism's underlying structure. In broader biological context, the platypus stands as a reminder that natural systems do not always conform to human expectations of order or simplicity. Its survival across millions of years of environmental change demonstrates the effectiveness of specialized adaptation within stable ecological niches. Within Australian river systems, it continues to fulfill its ecological role as both predator and participant in freshwater food webs, contributing to the balance of aquatic ecosystems. Its presence also reflects the long continuity of evolutionary history in regions shaped by isolation and shifting climates. As freshwater habitats evolve under modern pressures, the species remains closely tied to the integrity of river corridors, where water quality and flow patterns determine long-term viability. Continued scientific observation of its behavior and genetics provides valuable insight into how ancient lineages persist alongside more recently evolved mammals, reinforcing its role as a living reference point for evolutionary biology. Modern scientific inquiry treats the platypus not as an anomaly, but as an example of evolutionary possibility. Its existence expands understanding of how life can diversify under different environmental constraints and historical conditions. Each aspect of its biology contributes to a broader picture of evolutionary innovation, where familiar categories are insufficient to capture the full range of natural variation. In this sense, the platypus remains an enduring illustration of biological complexity that resists reduction to simple explanation. Ongoing research continues to refine how scientists interpret its traits, particularly as new genetic and physiological data become available. Each discovery adds further depth to the understanding of monotreme evolution and highlights connections between ancient vertebrate lineages and modern mammalian systems. Rather than fitting into a fixed framework, the species encourages continuous revision of biological models. Its persistence across vast spans of geological time reinforces the idea that survival is often linked to adaptability within stable ecological niches. As our journey through the quiet river comes to a close, picture the platypus slipping beneath the water's surface, leaving only gentle ripples behind. The river slows into stillness once more, reflecting the fading light of evening. There is a quiet wonder in this world, where nature creates the unexpected, yet everything still feels peaceful and balanced. Let the gentle sensations of peace and calmness settle through your body and mind. Feel your thoughts slow, your breathing deepen, and your body fully relax. If you enjoyed this relaxing moment, please consider liking, subscribing, or sharing your thoughts below. You can also turn on the notification so you don't miss any of our future journeys together. But for now, let your body relax, release all tension, and allow your mind to drift gently into rest. Good night and sleep well.